How do scientists identify a virus when the cause of pneumonia is completely unknown?
It sounds like researchers would need a way to search without already knowing what they are trying to find. But how can they recognize something unfamiliar?
That was the challenge described in this paper. In December 2019, a cluster of patients with pneumonia of unknown cause was linked to a seafood wholesale market in Wuhan, China. Researchers needed to investigate what was behind these cases.
So the first problem was not just understanding the illness, but discovering the cause itself.
Exactly. The researchers analyzed samples from patients and discovered a previously unknown betacoronavirus. The paper is titled "A Novel Coronavirus from Patients with Pneumonia in China, 2019, and it describes the identification of this new virus.
Was the key surprise that the virus was not one of the coronaviruses already known to infect humans?
Yes, the study showed that researchers had found a new coronavirus and then examined its characteristics and relationship to other members of the coronavirus family.
Why is identifying the virus such an important first step?
Because when the cause of a disease is unknown, researchers cannot properly study that cause until they have evidence about what it is. This work changed an unexplained pneumonia cluster into a defined scientific subject. The researchers faced a difficult question. How can you find a virus without starting from a list of known possibilities?
If they only searched for familiar viruses, they might miss something completely new.
That is why they used unbiased sequencing on samples from patients with pneumonia. This method examines genetic information without assuming a specific virus is responsible from the beginning.
So instead of asking whether a sample contains one known virus, they looked broadly for genetic clues.
Exactly. This approach helped reveal a previously unknown betacoronavirus in the patient samples.
But finding genetic clues alone does not seem enough to fully study a virus. What happened next?
The researchers used human airway epithelial cells to isolate the novel coronavirus. In simple terms, isolation means obtaining the virus in a form that allows researchers to examine it more directly.
Why does that make a difference?
Because it provided material for further examination of the virus itself. Together, the sequencing results and the isolation of the virus supported the identification of this new coronavirus.
So the method was designed to answer the basic discovery question: what was causing these pneumonia cases?
Yes. The focus of this study was discovering and describing the virus responsible for the cases being investigated. After identifying the virus, the researchers asked another important question. Where does this new virus belong among known coronaviruses?
Why does its position in the coronavirus family matter?
Classification helps scientists describe relationships between viruses. The paper reported that 2019 nCoV formed a clade within the subgenus Sarbecovirus of the Orthocoronavirini subfamily.
Those terms are difficult to understand. What do they mean in a simpler way?
Think of a family tree. A clade is a group of organisms that share a related branch. The researchers were placing this newly identified virus within the larger coronavirus family based on the evidence they examined.
Does being related to other coronaviruses mean it was the same as SARS-CoV or MERS-CoV?
No. The paper specifically stated that 2019 nCoV was different from both SARS-CoV and MERS-CoV. It was identified as the seventh member of the family of coronaviruses known to infect humans.
So the researchers were showing both what was new and how it connected to existing knowledge.
Exactly. A new discovery becomes easier to study when scientists can describe its place in a known framework.
And that classification came after the virus was identified from the patient samples.
Yes, the study connected detection, isolation, and classification to describe this previously unknown coronavirus. The central finding of this research was the discovery of a previously unknown coronavirus from patients with pneumonia of unknown cause.
What evidence supported that discovery?
The researchers found the virus through unbiased sequencing of patient samples and used human airway epithelial cells to isolate the novel coronavirus. These steps allowed them to examine and classify the virus.
So the conclusion was based on a process rather than a single observation.
Correct. Genetic information revealed something unexpected, and isolation provided a way to study the virus more directly. The evidence supported the identification of a new human coronavirus.
Did this paper claim that discovering the virus answered every question about the pneumonia cases?
No, the paper focused on identifying and characterizing the virus. It also stated that enhanced surveillance and further investigation were ongoing.
That distinction is important. Finding the cause is a major step, but it is not the same as understanding every aspect of it.
Exactly. The value of this study was creating a clear scientific foundation. Researchers moved from an unknown cause of pneumonia to a known virus that could be examined using scientific methods.
So the discovery itself was the main result.
Yes. The paper provided evidence that a new coronavirus existed, showed its relationship within the coronavirus family, and established a starting point for continued investigation. The most important lesson from this paper is that science can make progress even when the first question is simply, what is causing this?
Because before researchers can study a new disease cause, they first need to identify what they are dealing with.
Exactly. In this study, researchers did not begin with a confirmed answer. They used unbiased sequencing to search broadly, isolated the virus, and then classified it among known coronaviruses.
So the process moved from uncertainty to evidence, step by step.
Yes. The discovery of 2019 nCoV transformed an unknown pneumonia cause into an identified virus that researchers could investigate.
What makes that discovery especially meaningful?
It provided a shared scientific starting point. Once researchers know the identity and classification of a virus, they can ask more specific questions about it using evidence rather than speculation.
The paper is really about the moment when something unknown becomes something that can be studied.
That is exactly the key insight. The researchers demonstrated how careful analysis of patient samples can reveal a previously unknown coronavirus and place it within the broader coronavirus family.
So the breakthrough was not only finding a new virus, but creating the knowledge needed to understand that discovery.
Yes. When scientists encounter an unfamiliar biological event, reliable identification is the foundation that allows further investigation to begin. This foundation helps turn uncertainty into a clearer scientific path for future questions.